नहीं होता। My research topic is biochemical profile of freshwater fishes from Nakana Lake, Dhule. Fishes are quite different from the other animals' food source because it provides high quality proteins, calories, all amino acids and some vitamins, that is A, D, K. It is palatable. Objective of my study is to estimate the dietary values of fishes, to mention variety in their nutritional combinations, to detect dietetic significance in between commercially important and small indigenous fishes. The study area, Nakana Lake is a man-made lake which built on the Panjra River from British period. It is placed in the West Khandesh, belong to North Maharashtra region. Actually, the main purpose of behind the construction of this lake, the supply of the water to the Dhule city, then irrigation to nearby farms, and before 2 to 3 decades, it is used for aquaculture. The map showing study area. During the research tenure, variety of fishes has been observed. Total 20 fish species were reported for the biochemical analysis, which is distributed in 5 orders. Appropriate amount of entire body weight tissue of fishes was taken. Tissue was homogenized, and the supernatant was used for the estimation of the glycogen, lipid, protein and the moisture. 6 observations were made and mean and standard deviation were calculated. The study tenure is February 2016 to January 2018. Glycogen, protein, lipid and moisture determined by the Miller, Lowry, Jayaraman and anonymous method. These are the fish species which are observed from the Nakana Lake. Now the estimated values are put into the table number 1. The highlighted colors are used for the glycogen, protein, lipid and moisture. In the present investigation, the higher glycogen content is present in Chana punctata, that is 6.55, then Cyprinus scarpio, that is 6.20, and the Mustas emballus armatus, that is 6.5. The while minimum in Parambesis ranga, Rasbora doniconeus and the Puntius saphor. The higher content of the protein is occurred in Tenulicia ilicia, that is 22.85, then Mustas emballus armatus, that is 21.15. In a Chana punctata, the content of the protein is 20 to 20.15, and the remaining the least content of the protein occur in a Mistus blickeri, Heprotmistus fossilis and Glossogurius guris. The maximum percentage of lipid shown in a Tenulicia ilicia again, that is 9.45, and it is followed by Mustas emballus armatus, that is 7.90, and after that Chana punctata, that is 5.90, and the least count of the lipid content in a Chandanama, Oreochromis mosambicus and Umpac bonaculatus. The estimated percentage of the moisture ranges in 83.20 to 45.10 in fish, Parambesis ranga and Mustas emballus armatus. In nutshell, Tenulicia ilicia, Mustas emballus armatus and Chana punctata are the nutritious fishes are as compared to other 17 fish species. Tenulicia ilicia is a small indigenous fish. Mustas emballus armatus is a commercially important fish, and the Chana punctata is exotic fish. After that, this biochemical estimation, Nakana Lake, no doubt Nakana Lake is harbor the very nutritious and enormous fishes, but some anthropogenic activities are happen here. Due to the these anthropogenic activities, the content of the combination of the biochemical analysis of the fishes are changed. At the time of Ganesh Chaturdashi and Vijayadashmi, the huge idol of the Lord Ganesha and Ma Durga immersed into the Nakana lake. The crowd is generally, fish quality depends upon the biochemical composition of the whole body, which is revealing the decline energy reserves and storage of the energy. Hence, their assessment of nutritional values is related to the physico-chemical characteristic of lake water, size of the fishes, gender, physiological activities, and feeding habits, and other influencing factors are composition of the food, genome, and particularly on environment. No doubt, Nakana Lake harbors commercially important as well as small indigenous fishes, but some periodic anthropogenic activities are happened here. From few years, increase the event of the immersion of the idol into the lake, dyes and colors of the idol change the chemical composition of the water. Due to siltation, reduce the water storage capacity, which affect the physiological activity of the fishes, and it vanished the spawning ground. Obviously, all these changes altering the biological composition of the fishes. In future, the current study compared with seasonal variation should be continued. I grateful to Indian Science Congress, RTM University Nagpur, referral committee, my colleagues, helping hands, and each and every audience of my presentation. Thank you, sir.
Now, this talk is open for question from the audience.
Yes, sir.
What is the size of Tenolisa elisa in this lake? Lake size is not big, I think. What is the size of this lake?
Sir.
You have not mentioned the size in your slide.
Yes, sir. Yes, sir. Tenolisa elisa is a small indigenous fish.
What is the size of this lake? First, you answer that question.
Okay, sorry, sir.
What is the size of that lake? 100 hectare?
No.
500 hectare?
I think 200. 200
hectare.
230 to 35.
Okay, 250 hectare at present.
Okay.
And what is the size of Tenolisa elisa in this lake?
Sir, maximum of it is maximum 2.5 to 3 centimeter.
That's why I am asking this question, because it's an anadromous fish.
Yes,
and
sir.
it comes from sea to reproduce in fresh water.
Yes.
Tenolisa elisa. But your size is too low. 2.5
Yes. Yes,
2.5 centimeter to 3 centimeter.
Yes,
if it is correct, you have identified correctly that is Tenolisa elisa.
Yes, sir. Yes, sir.
Because a lot of fishes are just similar to
Yes, sir.
elisa in this size. In this size.
No, sir. These species are identified by GDSI. GDSI.
Oh, GDSI find there also in
Yes,
this lake.
Yes,
okay.
sir.
But the size is not increasing. This is only this level.
Yes, sir.
Okay. No, that is okay. Because that is freshwater level. Because if it will increase from this size, it will take at least 1 year to grow.
Okay.
More than this size. Okay,
sir. Thank you, sir. Sir, can I? Ma'am, yeah. Thank you.
Thank you. Then the next. Dr. Fulgain Kumar.
My study is that histopathological assessment of chronic exposure of dichlorvas on freshwater Channa punctatus fish. Dichlorvas, that is a chemical, is an organophosphate compounds, and it is used in controlling household and public health and effective against mushroom, flies, caterpillars and many others. And they causes cellular toxification process may be different, present in more or general well-known situation of oxidative stress due to the production of free radicals. The free radicals that causes damage surrounding the cytoplasm and other parts of the cells. It was found that after chronic exposure, damage values were highest at all the concentration in the histopathological tissues. The present study was planned to evaluate histopathological assessment of chronic exposure of dichlorovas, the freshwater fish Channa punctatus. My test organism is freshwater fish, that is a Channa punctatus. Chemical, that is dichlorovas. Determination of LC50 is done by probit analysis methods, that is 1.26, and sublethal doses, that is a 1/4 of the LC50 and 1/8 of the LC50, 1/12 of the LC50 that expose in different concentration. After that, the tissue, after exposure of the chemicals, the histophysical observation were done in different tissue like gills, liver, kidney. So this is the common procedures that is done for microtomy process. First of all, we fix the tissue in the fixative, that is a formalin. After that, it washed and then dehydrates. After dehydration, yes sir. Okay. And observation, this is the control gill tissue. After exposure 21 days, damage in the primary and secondary leveling and vascularization also take place. This is the picture of control tissue of kidney, and it's the after 21 days exposure, damage in different tissues, liver control tissue and liver after chronic days. There are damage in hepatocytes and other sites also. Dichlorovas has not been extensively monitored in most environmental media. It has detected in very low concentration in outdoor air, but is detected in higher concentration in indoor air associated with its use as fumigants, pesticide in homes or during conventional exposure, manufacturing and production facilities. It has been detected in drinking waters, and no environmental monitoring data were found for dichlorovas in surface or groundwater or in soil and sediments. Dichlorovas has been detected infrequently and never at concentration of caution in both raw and processed food items. Dichlorovas has been identified in at least 3 of these current farmers hazardous waste sites have been proposed. In present study, exposure of chronic concentration of dichlorovas to fish specimen induce gill, liver and kidney damage, indicating histopathological toxicity properties of dichlorovas to aquatic organism. Further, there was variability in gills, liver and kidney damage between the two tissues of the specimen exposed to the pesticides. Conclusion: the geotropic potential of chronic concentration of dichlorovas found in this investigation suggested a serious concern towards its potential danger to aquatic organism and its use in agriculture practices. However, further studies are needed to explore biological consequences of histopathological damage in Channa punctatus after dichlorovas exposure. Thank you.
Any questions?
No question.
Thank you. Thank you.
Next is Ganesh. Please come that time.
Open Ganesh Chandra. Okay. Which one is that? Which one? I have to press this one, na? Just tell me. Okay. Okay. Respected chairman, co-chairman, and my senior colleagues present in the audience. This is my presentation. Hilsa Indian Side Fisheries in downstream Namda estuary. Decadal analysis of fisher livelihood. Okay, this one. Okay, thank you, sir. So this decadal analysis, it's a totally primary data collected on 2 occasions in 2008-9 and 2018-19 period. We have selected the persons, fishers from different age group. It's a hierarchy age group, 60 plus, then 55 plus, then 45 plus, 35 plus, and 25 plus. These groups we have taken in our focus group discussion, and after that this data has come up, and I am showing those data only. Why Narmada estuary is important? After Sardar Sarovar Reservoir, this 162 kilometer area of river, it is the downstream area, and in this downstream area, if we can see, there are 2 species which is very important. From Bharuch town to lower side to up to the sea mouth, it is Hilsa, and upper means dam to Bharuch town, it is Macrobachinum rogerbergi, the freshwater scampi. 2 species are important here, and this whole period, this study is also very important because this study covers the period pre-Sardar Sarovar project, Sardar Sarovar project time, and after Sardar Sarovar dam has got the height of 138 meter. It has completed it just 3 years back in 2017. I was there luckily on that day I was also there because of some invitation from Sardar Sarovar Narmada Nigam Limited. So this study covers the 5 period, means 5 decades study, and I will just show those things. And why it is important, I have already discussed. And this is Hilsa fishing is mainly done in the 45 days period of monsoon. And our maximum catch is during the high tide of new moon or no moon period of the month. And mainly the fiber craft at present used by the fishers, and 5 fishers were involved in 1 boat. So, and if you see the catch, how the catch has declined after the Sardar Sarovar dam, after the establishment of Sardar Sarovar Dam, it was just started from 1994, and you see from 1994 to 2000, how this had changed. 2010 period, that time the height was only 121 meter. Now the height has been increased to 138 meter. Again, I have already talked about that, and just the parameter what I am covering in this: fisher household and population, fishing intensity, fishing days, employment from fisheries, fisheries requisite like craft and gear, then fishing practice and season with a special reference to Hilsa, and fish catch in terms of yield per boat.
So please skip to the results, sir. You're just
No, it is result only. Methodology
Faster.
is just over. And why this is also important, during this 5 decade, this Jhanor has come to the zone 2. Zone 2 means the tidal freshwater zone, and which is up to Bharuch was freshwater zone, which has now gone to the freshwater zone is only up to Sisodra. Okay. Now, why this species is important? In monsoon catch, if you see in zone 3, it is 98% catch is Hilsa, and in zone 2, it is 47.73% is catch of Hilsa only. Now I come to the result one by one. The fisher population, if you see how they have increased over the years, 5 decades. This is highly increased in Jhanor. Jhanor is especially the belt where the tidal zone starts, and also the Bharvut. Bharvut is the zone where the trade going on. Then the fisher household, if you see the fisher household, not in freshwater zone. Freshwater zone is almost stagnant, but you see in this brackish water zone, it has grown massively. This fisher household also increased. It is 242% population increase in 4 decades and 135% household increase in last 4 decades. And if you see the employment from fisheries, it is drastically down from 1978 to 2018 period. But if you see the linear trend of 2018, it is almost stagnant at near about 40 to 50% level. Then the craft has also, the craft as well as the gear, both have changed. Now at present it is nylon gear, and in place of cotton it is nylon gear only, and craft has come from tin to. And if you see the total fishing days, how this has declined over the 5 decades. If you see the freshwater area as well as brackish water area, it has the, except Sakarpura, because Sakarpura is only fishing for hilsa, no other fishes. While in other area, all other fishes are fishing is done. If you come to the fishing days for hilsa, it is also slightly declined in 2018 in Bharbut region, because one reason is there for this decline. The Sardar Sarovar Narmada Nigam Limited is only releasing 600 cubic water per day at present, which was earlier more than 2000. We have already, they have given a consultancy, this is under a consultancy project. So they have given also for consultancy for e-flow. We have suggested for increase this to 2000 cubic per day at present. So most probably the Water Resource Ministry of Government of India will take action on that. Then come to fishing hours. You see the fishing hours is almost same from Jhanor, the brackish water zone. It is almost 12 hours at present from last 4 decades. Last 4 decades. It has no change, but it has changed in freshwater zone. Then fishing stretch, it is also little bit increasing. Every 10 years it is increasing because hilsa catch has dwindled, hilsa catch has come down. And if you see the hilsa catch per boat, while it was increasing during 1978, but the trend line of this, you see just the trend line of 2018, it has drastically gone down. And if you say the gender dimension of hilsa fishing, the initial arrangement is male dominated, catching is totally male dominated. But after the bank, when the hilsa comes to the bank, it is totally female dominated. The money is kept by female. And this is one area I can tell you the fisher women empowerment is more in Gujarat, because they are keeping all money. They are keeping all money. Not like in Uttar Pradesh and Bihar, where the fisherman is keeping all money and they are utilizing for liquor and other things. Here the whole things are going for their family benefit. It is also one of the most important area. Value chain is also important here, because in not like Calcutta where hilsa is any type of hilsa you can purchase at 1500 rupees kg or 2000 rupees kg. In Gujarat it is different. Gujarat 1.5 kilo size of female hilsa is almost 2500 plus, because single nug hilsa is sold there. Single fish, not as kilo. In Gujarat, it is totally, a grading is different. Sir, this is last slide, sir. Last slide only. So, decadal trend of hilsa catch shows declining trend. The fishing stretch increased from 5 km in 1978 to 30 km in 2018, and drastic catch. This is our 2 study in 2008 and to 10 consultancy project, and again 2018 to 20 consultancy project. What we found, drastically down of catch per unit effort. This is now just 17 kg per boat, which was earlier 74 kg in just 10 years back. Within 10 years, it has drastically gone down. It is one of the reasons the livelihood has gone down, drastically changed of all the fishers. Again, the Bharbut and Mehegam has emerged as the main landing center in place of Bharuch and Bharbut earlier. The fishing season has comparatively extended in lower stretch from July to September as compared to upper stretches, where it is only hilsa is not at all available. The negligible catch in upper stretches. And income from hilsa fisheries has declined sharply after 98 with the construction of Sardar Sarovar Dam. Adversely impacted the livelihood of the fisherman community. Thank you very much. This is one lady which is carrying her hilsa for their home purpose. The rest all is sold. One is for their own cooking purpose. Thank you very much.
Well, Ganesh is from Sifri Barakpur. Okay? If you have any questions, any inquiry, any information, okay? This session is wide open for the audience. If you have anything to ask, you can, beyond even this talk. Okay?
Nobody is interested in law. Sir, you can ask. You are the senior most professor here. Dr. Raghunath is also here.
No one is interested to know more. So thank you.
Thank you.
This is the pathetic stage. Very good presentations are being lost at the time. And now everybody wants to know more, but because of the time, I really appreciate the very sincere and broad work that you have done, sir. But it should be presented in a larger forum and in the right time.
That's
and
fine.
everybody now, they want to present and they want to get the certificate. Sure.
Thank you very much.
Thank you very much, sir,
Thank you,
for
sir.
the presentation. Next, Badal Kumar Das is here.
This one? Yeah.
Yes, sir.
I have presented only graphs.
These are first slides. This one? Pointer.
That's what you didn't tell anything. I was expecting you.
So good evening, everyone. So respected chairperson, co-chairperson, distinguished scientists of this session, ladies and gentlemen. I am Dr. Badal Kumar Das from UN Autonomous College, Science and Technology. Now presenting the topic, that is mortality studies in pestiferous Levicalis salti using neem products as plant-derived molluscides. So the background of this particular work is Levicalis salti is the most destructive invasive molluscan pest present in our locality, damaging agri-horticultural crops and ornamental plants. For that particular species, the molluscan species must be controlled, for which normally the chemicals are going to be used, which are causing environmental pollution as well as death to the non-target organisms. So this is just a particular work to search for eco-friendly, cheaper and readily available renewable alternative material for controlling that particular snake, for which we have used the plant products as botanical molluscides. So this is the Levicalis salti, which is causing the damaging effect. The objectives, that means the selection of Azadiracta indica, the plant material as a suitable molluscide agent, the standardization of the methodology in the laboratory condition, extracting the phytoproducts from that particular plant, and studies on the impact of selected plant-derived molluscides and the mortality rate of the species, examining the selected materials, particularly of different categories for study of the mortality, and finally to analyze the results of the mortality data, depending whether that is related to the concentration as well as the duration of the treatment of that particular species using the phytoproducts having the toxic ingredient. This is the exact species, Levicalis salti, and selecting Azadiracta indica, which is having the respective type of agromedicinal property and the biopesticide, and it can be very well having the plant material with internal chemical defense, making the neem to be free from pest attack, and for which it is used as an efficient pest control strategy. And the functional ingredients of this particular neem is therapeutic, anti-inflammatory properties, antibiotic, immunomodulatory. It can be used alone or in combination with other herbs. And this is the toxic component of the neem, which is known as azadiractin, which is having 0.1 to 0.3% in the seeds, and the respective toxic constituent is very low in its toxic potential to the mammals. It is having the complex tetra or triterpenoid limonoids C35H44O6 with bicidial efficacy. And it is similar to that of ecdation, which is influencing the metamorphosis in case of insects. Other triterpenoids are also there, which are having the bioinsecticide spots. And the preparation of the plant material, that means they have been collected from the respective sources, that means the seed, bark, board, leaves, etc. They are that cut into small pieces and air dried or sun dried, and they are use the electric bender and airtight containers. The powder is going to be stored. And the methodology we have used, there are so many methods, but first of all, to have the efficiency of that particular material, we have tested the dusting method, then spraying, and finally we have standardized the animal immersion technique, which the entire animal is going to be immersed in that particular solution or the extract, and for which the toxicity is going to be resulted because of contact one. And this aqueous preparation is restrictively utilized for 24 hours of duration in its treatment. And additionally, this entire process is also compared with the distilled water as the control environment. 5 different concentrations have been taken into consideration: 500 ppm, 1000 ppm, 1500, 2000, 2500, and the death has been completely analyzed after poking the needle in a particular time period of recovery. And the observations in the dusting method, the neem bod and neem seed has been found to be completely effective, which is causing slime secretion and resulting in the death of that particular animal. They are highly toxic. And neem bark and neem leaf powder, they are not going to be so much toxic, and they are moderately or completely less toxic in the dusting method. These are the results in the dusting method by which the animal is going to be killed in that particular dusting process. But we have standardized the total animal cell immersion method. Here, aqueous extract of neem powder and as well as the aqueous extract of the neem seed powder, they are causing the immediate slime secretion on the body surface, rapid movement, and escaping from the aqueous toxic environment, and twisting of the body, and sometimes the movement stopped and body becomes paralyzed and the protruded mouth with the tentacle, and the animal is dying. And this is highly toxic. At the same type of observation is also going to be found in case of seed powder. But in case of aqueous extract of neem bark and neem leaf, the entire survival is beyond also 24 hours. It is also surviving beyond 24 hours. But the exact observation is within 24 hours. These are the results, and the observations are like this, and we have taken 3 to 5 specimens in a particular replicate. This shows the total slog mortality on total immersion using aqueous neem bod and seed solution. This is the neem bod experimental observation, and this is the seed solution, and the animal is dying in this particular procedure. And this is the result of the neem bark in aqueous solution and the leaf solution. Here you can see the leaf solution treatment is still giving survivability of that particular animal, and it is coming outside and trying to come outside. And these are the graphical representations by which this is the concentration and duration of survivability. When the concentration is increasing, the duration of survivability of that particular animal is just gradually declining. And if the duration of exposure is going to be increased, naturally the percentage of mortality is also going to be increased. This is also same. This is in case of neem bark. And this is the graph which is showing the neem seed, the same type of trend. And in case of the neem leaf, which is completely going to be less effective, and the respective duration of exposure is much higher, but still then it is not dying. And finally, the bod has been proved to be most effective, and the respective contribution in form of concentration is also going to increase, as well as the respective survivability is also declining. So duration of exposure is also going to be increasing, and the percentage of mortality is also increasing. Here, this is the complete graph which is going to give you the idea about the concentration of 4 different categories: neem bod, neem seed, neem bark, and neem leaf, which are just giving you the 100% mortality. Particularly neem bod and neem seed, they are going to give you 100% mortality, whereas leaf is going to be less toxic. But coming to the discussion, my objective was to finalize the phytomolecular from the particular neem plant to give you the potential towards the death of the Laeviculaceae, and the contributions made by scientists like Abu Bakr, that it is going to be completely controlling the mode of action, which is going to be vital for limiting the toxicity of respective non-target organisms. That is the one point. And another aqueous extract, which is going to be completely contributed by the Henderson 1968, Wilkins, and thus myself also. We have already observed the contact toxicity is going to cause slime secretion and the death of that animal out of desiccation. And water extract in case of neem berries, when tested in poultry birds even, within 7 days, it is also going to show some sorts of toxicity. And the molluscidal effect in case of Laeviculaceae and Achatina folica, whenever the silica particles are coated with botanical pesticides like neem, currant, and tobacco, neem has given some sorts of effect, where within 24 hours there is the observation of inactivation, body fluid loss, and the death out of desiccation. And the effect of the neem leaf in case of Achatina also, the leaf is going to be completely proved as the destructive material which is going to kill the snail. The molecular property in case of Limnaea acuminata Endoplanurobis justus is going to observe the respective increase in form of either the time or the concentration, either the time or the dose dependent, it is increasing. And the present investigation is having the result that both the bod and seed, they have been proved to be completely influential over the bark and leaf whenever they have been used in form of aqueous extract solution. And this is exactly increasing, and the duration of exposure is also related to that one. And my concluding remark is that mortality studies is going to be the basic tool to evaluate the toxicity whenever considering the lethal and sublethal concentrations, particularly using either any kind of chemical molecide or the plant molicides. And in present investigation, what I have already told, that is exactly the intoxicated environment or the complete controlled environment. It is increasing concentration as well as the duration of treatment. So the future prospects are estimation of the exact lethal concentration to find out the phytophthorus neem, which is going to be eco-friendly or cost-effective alternative to the chemical molicides, and the toxic potential has to be completely evaluated prior to its implementation and standardization of the technique for the respective integrated pest management practice. And for the rural farmers, they are going to be completely implement the respective neem plant products in their locality to protect the crops and plants, particularly in our locality even. This is a suggestion for the respective individuals. And I am very much thankful to the President, Governing Body of Union Autonomous College and the Principal for giving me this opportunity. And these are the respective references.
Thank you, Dr. Badal Kumar Das.
Thank you.
You are running at a speed of more than 100 kilometers. Very quick, but you know, we have time constraint. It is around 8:30 today. Okay. But anyway, your talk is very informative, and in spite of high speed and quick succession of words, things are audible. Okay. So, any question from the audience? There is no question, so thank you very much.
So, thank you, sir.
And last presentation is there. Just have a patience for 5-10 minutes more. Which one? Dr. Abhay Shelke. Okay. Please come, gentlemen, and do a favor on all of us. Okay. Just conclude your talk within 5 minutes.
Yes, sir. Thank you. PPT.
Thank you.
Dr. Abhay. 2 number. No. He puts in. Respected Chair, respected dignitaries, myself Dr. Abhay Shelke, Department of Zoology, PP Art Science Commerce College, Chalisgaon, KBC, North Maharashtra University, Jalgaon. Title of my paper is Evaluation of Oxidative Stress Caused by Silver Nanoparticles in Zebrafish Dinoario. Objective was to study the chronic effect of silver nanoparticles on antioxidant enzyme like catalase, superoxide dismutase, glutathione peroxidase, reduced glutathione activity, lactate dehydrogenase, and lipid peroxidation activities in liver, gill, and muscle tissues in adult zebrafish Dinoario. One objective was to evaluate the oxidative stress of silver nanoparticles in the zebrafish Dinoario, to study the oxidative stress related behavior of Dinoario in chronic exposure of silver nanoparticles. Silver nanoparticle suspension, preparation, characterization. Silver nanoparticles powder which having 60 nanometer diameter coated with polyvinyl pyridine with a purity of 99.5% was purchased from Indian Platinum Private Limited, Vileparle, West Mumbai. PUP coated silver nanoparticle was chosen as they should easily disperse in water. The morphology, average diameter and dispersion of silver nanoparticles were determined with the field emission scanning electron microscopy FESM at University Institute of Chemical Technology, KBC, North Maharashtra University, Jalgaon. Powder sample of silver nanoparticle was used for electron dispersion X-ray spectroscopy analysis and XR. Yes, ma'am. So these are the images of characterization of silver nanoparticles. In the acute toxicity study, adult fish Dinoario. Yes. This is the experimental fish, zebrafish Dinoario. This is the behavior study that I have observed during the experimental. These are the methodologies I have used for this study. These are the results. Catalase activity in zebrafish Dinoario exposed to silver nanoparticles during 21 days, and you can observe this. There is a decrease in the catalase activities in the liver, gill, muscle tissues of zebrafish Dinoario. These are the graphical representation. It indicates clearly there is a decrease in the catalase activity. SOD, that is superoxide dismutase activity, dinurerio exposed to silver nanoparticles. There is also increase in the SOD in the liver, gill, muscle tissues of zebrafish dinurerio. These are the graphical representation, it clearly indicates that. GPX, that is the glutathione peroxidase activity in the zebrafish dinurerio exposed to silver nanoparticles. In the tabular, you can see that there is a increase in the GPX. This is the graphical representation. Reduced glutathione activity in a dinurerio, you can see there is a decrease in the reduced glutathione activities in the liver, gill, muscles of dinurerio. There is a significant decline in the reduced glutathione activity. Lactate dehydrogenase activity in dinurerio, there is also increase in the LDH in the liver, gill, muscles of dinurerio. This is the graphical representation. Lipid peroxidation activity in the dinurerio exposed to silver nanoparticles. There is also increase in the lipid peroxidation activities. This is the graphical representation. These are the results.
Sir, in a nutshell, I request whether the nanoparticle is the side showing the positive or negative impact.
It's a negative impact, sir.
That's what you want.
Yes, yes. It's too much negative impact, sir.
My question is, in what way this silver nanoparticle is related to zebra?
Yes, sir.
Is it present in the water?
Sir, coming era is nanotechnology era, and when the nanoparticle based product will come in the market, then all these scraps will be come into freshwater reservoir, and that directly come into food chain and food web, and in coming future era we will be the part of this.
Okay, might be you are the first person to say about it.
Yes, sir.
Any more questions? So thank you, Dr. Abhay.
Thank you, sir.
And thank you, audience. Thank you.
Thank you, sir.
Persons
Thank you.
still available here.
Thank you, sir. Thank you.
Dr. M.G. Raghunathan, Dr. Ganesh, Professor A.M. Saxena. Only 3 persons left in the audience. And of course, my team, Jayanti and this lady, what's the good name? Kareem, you? Saloni, Tiwari. Okay, wonderful. Then, madam? Mangala Thakre, all Mangala, Saloni and Kareem, I thank you very much. Jayanti,
Thanks to all.
I am grateful to you. And the last 3 persons left with us, M.G. Raghunathan sahab, camera persons, my friend, what's the good name, sir? Girish, and this photographer, Nishikant. Oh, okay, I am, thank you. Thank you very much.
नहीं सर थैंक यू थैंक यू
वही तो कह रहे हैं मोमेंट के बाद सर
Now I request Jaiswal sir to felicitate Dr. Ulfat Jan ma'am. Now I request Jaiswal sir to felicitate Professor Yahya Bakhtiar sir.
भैया आइए जनाब तो सबसे इम्पोर्टेंट वाला है भाई भैया इनको ठंडी नहीं लगती तो क्या पहना है शॉल? थैंक यू यार। सेक्शन में ओरल होंगे ना? ओरल ठीक है, बोलो ओरल।
Now we are moving towards the oral presentation. For this session, we have 9 oral presentation. Dr. Professor S.P. Trivedi sir and Dr. J. Jayanti ma'am are the chairperson and co-chairperson for this session. Now I request chairperson and co-chairperson to proceed the session ahead.